Hello everyone, we are here in my kitchen. It's very late at night and we are going to talk about the endocrine system. So let's go ahead and get started. We will be going through, in this particular video, we're going to talk about general mechanisms, how hormones do what they do, and then we'll be breaking out into specific glands to talk about each of them in more detail. So I'm going to start with just giving you a reminder about different gland types that we have. We've seen this earlier in the semester, back in chapter 1. So with glands, there are two different types, exocrine and endocrine. And exocrine glands, let me just get my laser pointer here. Exocrine glands, like on the left, exocrine glands have a duct that leads to some sort of a surface. So these can secrete out of the gland directly. Endocrine glands, what we're going to be talking about in this chapter, endocrine glands are more like what's shown on the right. So there is no duct leading out to the surface. Instead, what happens is this gland produces hormones, and those hormones diffuse through the tissues, and they get taken up by the bloodstream via a capillary. So endocrine glands, the hormones are staying inside essentially, and they're just being transported through the blood. So when we use the word hormones, what do we mean by a hormone? A hormone is just a regulatory molecule, and it's generally transported by the blood. So there are several specific glands throughout the body that produce hormones, and we'll be looking at the major glands. For all of the hormones that are produced, keep in mind that each one has a specific target cell that it's going to act on. And the specificity of hormones just comes down to what sort of receptors can it bind to. So there are a lot of similarities with neurotransmitters in that sense. So what do hormones do? They do a lot of very important things in the body, but some general functions are to regulate metabolism. Hormones control how our cells are able to take up glucose or not take up glucose, so energy metabolism. They also influence growth of tissues and growth of organisms as a whole, and they also allow reproduction. So again, major organs that are involved in hormone production are shown in the diagram here, but keep in mind that even organs that are not listed as major endocrine glands, many other organs also produce hormones, just in smaller amounts. So we're just going to be focused on the major ones in this chapter. And to tie back in with the nervous system, what we've just come from in the course, you remember that we said the hypothalamus can produce some hormones, and they're stored in the pituitary gland. We're going to be seeing that in more detail today. But hormones that are produced by nerve cells are called neurohormones. And with that in mind, let's start by sort of comparing and contrasting. There are going to be a lot of similarities in this chapter with hormones, a lot of similarities back with neurotransmitters. So some of the things that they have in common, they're both very specific. They bind to specific receptors, and that allows them to target particular cells in terms of the actions that they cause. And always the receptor binding, that's the thing that initiates some sort of a change or some sort of a signaling cascade to take place inside of the target cell. With neurotransmitters and hormones, they both have to have a means of being deactivated, and oftentimes that will involve either taking back up the molecule or inactivating it, maybe cleaving it in half. So there are mechanisms in place to turn off the signal that's being sent. And then finally, keep in mind that some of the molecules we've already seen as neurotransmitters, some of those are going to show up again, and they're going to be acting as hormones. A good example of this is norepinephrine. So we've seen this as a neurotransmitter, but now in this chapter we're going to be seeing it acting as a hormone. So sometimes the same molecule can be used in two different ways in the body. Hormones can be classified a couple of different ways. We can classify them based on their chemical structure, and based on that classification there are four major categories. Amines, we've seen these again before. These were neurotransmitters in the last couple of chapters we've been through. Amines are just derived from amino acids, tyrosine and tryptophan specifically. Polypeptides and proteins, these are larger molecules, so this consists of multiple amino acids connected together. Again, we've seen examples of this as well. Antidiuretic hormone, we've mentioned that in the past. We'll be seeing growth hormone and insulin in this chapter in more detail. And then glycoproteins. So glycoproteins, if you'll recall, this is a protein with a carbohydrate attached to it. Glycoproteins, a good example of a glycoprotein that is a hormone is, well, there are a couple listed on the slide, but any of the gonadotropic hormones that we'll be going over are glycoproteins. And then finally on the list, we've got steroids, and steroids have that distinctive ring structure. This was one of a type of lipid that we saw back when we were talking about different types of chemical molecules. So we can classify based on chemical structure. We could alternatively classify hormones based on their action, how they do what they do. So looking at it this way around, there are some hormones that can cross the plasma membrane, and there are others that cannot cross the plasma membrane. And that's a big difference, being able to cross that membrane or not. That leads to different approaches in terms of how the hormone does what it does. So if we're talking about polar hormones, so big polar molecules, these generally cannot cross through the plasma membrane, right, because it's lipid. And so this means that these hormones have to bind on the cell surface. So the receptor for the hormone is on the plasma membrane, it's on the cell surface, and receptor binding then initiates some sort of a cascade of events inside of the cell. Okay, the nonpolar hormones, these are often called lipophilic. They like to be with lipids, and these are the types of hormones that can just diffuse directly through the plasma membrane. They're able to cross, and this means that their receptors, they don't have to be on the outside of the cell, they can actually be internal, they can be inside of the cell. And a lot of times these hormones end up influencing activities in the nucleus. They influence gene transcription almost directly. We'll see that in a little bit. So two very different means of action for hormones. In either case, regardless of which category we're talking about, when the hormone makes it to its target cell, there can be a couple of different ways the cell will respond. So it depends on how much hormone is present, how much is binding to receptors in the target cell, and it also depends on the combination of hormones. And with regards to different combinations of hormones, sometimes there can be multiple hormones that will act together to result in sort of the same consequence. A good example of this is epinephrine and norepinephrine. Both of these act to increase the heart rate, so that would be an example of a synergistic effect, a synergistic interaction between two hormones. Alternatively, we could have hormones that have opposing effects, and a good example of that we have mentioned already in the class earlier is insulin and glucagon. These have opposite effects in terms of blood glucose levels. So what insulin does is it promotes cells to take up glucose, and so that lowers the glucose concentration inside of the blood, whereas glucagon does just the opposite. It ends up promoting the breakdown of glycogen into glucose, and so that raises the blood glucose levels in the body. Okay, we are going to go through a number of different mechanisms. I just want to give you a warning before we go into it. Lots of different mechanisms coming up here, and it's good. Feel free to just pause the video between each one, sort of take a minute to absorb what is going on. Let's start with, this is a general overview regarding nonpolar hormones, so hormones that are able to cross through the plasma membrane. And in this case, I'm just going to point on the slide here. In this case, what happens is the hormone, which is shown in blue, the hormone starts off in the blood, and it's attached to some sort of a carrier protein that shuttles it around through the bloodstream. So this is circulating all throughout the body. And when this hormone makes it nearby its target cell, what's going to happen is dissociation of the hormone from the carrier protein. And then this is a lipophilic hormone, so it is capable of just crossing through the plasma membrane. It can go across, it will diffuse into the cell. And then inside of the cell, there's some sort of a receptor. So receptor binding will take place in the cytoplasm. And something to sort of clarify right here, and when you're reading about these sorts of receptors, you'll read of them being called nuclear hormone receptors. Why is it called a nuclear hormone receptor? It's hanging out in the cytoplasm. The reason that it is called a nuclear hormone receptor is because once the hormone binds, this whole structure becomes activated to go into the nucleus. So it's headed into the nucleus next. All right. This receptor that's shown in green, okay, so it's bound to a hormone now. The other end is capable of binding to DNA. So once it's in the nucleus, DNA binding will take place, and that results in this whole complex becoming active as a transcription factor. So that is actually going to turn on a gene that is in the DNA. So it binds to what would be called the promoter sequence for a gene, and it activates transcription. So here's some mRNA that's being produced, and then that mRNA, of course, can go and be translated, and a protein will be made from it. Okay, another category of lipophilic hormones are thyroid hormones. These are produced by the thyroid, and I just want to show you kind of a breakdown of how thyroid hormones do what they do. It's extremely similar. Okay, so thyroxine is one thyroid hormone. Thyroxine has 4 iodines, and so sometimes it's abbreviated T4. So T4 likewise starts off circulating in the bloodstream. And once it makes it to its target cell, it will dissociate from its carrier protein, and just like what we saw on the previous slide. It will cross over the plasma membrane, so now it's inside of the cell. Inside of the cell, one of those iodines gets removed, so T4 gets converted to T3, now just has 3 iodines, and T3 can bind to a receptor. Okay, so there's a binding protein, and that binding protein, once it is bound, that whole structure will go into the nucleus, or rather, I'm sorry, the binding protein carries T3 to the nucleus, and then T3 goes inside of the nucleus, binds its receptor inside of the nucleus. So that's the thing that's different in this case, particularly different from on the last slide. Here the receptor is in the nucleus, and binding of T3 causes the receptor protein to become an active transcription factor. So extremely similar. The receptor protein in this case, it starts off already bound to the DNA, but it's not active as a transcription factor until T3 binds. So that's those are 2 mechanisms, examples of nonpolar hormones and the way that they work. So they're just able to go straight inside of the cell and bind to a receptor inside of the cell. In contrast to that, we can talk about hormones that are polar. So if it's a polar molecule, it's not going to be able to cross the plasma membrane unless it had some sort of special transport mechanism in place. But in general, polar hormones bind to the surface of the plasma membrane. There's a receptor embedded in the plasma membrane. And that's going to, binding is going to initiate some sort of a second messenger system inside of the cell. Second messengers, we've mentioned these before in passing. We'll see them in a lot more detail now. We're going to look at 3 mechanisms. So again, take your time as you go through this. Take it one at a time. Adenylate cyclase, this is the CAMP messenger system, cyclic AMP, and we'll look at these other 2 as well. So we've got one slide for each of these different second messenger systems. All right, so polar hormones. Okay, first example. All right, so hormones such as epinephrine and norepinephrine, these are good examples of hormones that cannot cross the plasma membrane. So what they will do is bind to a receptor on the cell surface. The receptor is in orange right here. That receptor is associated with a G protein. G proteins, remember what they do, and when they're activated, they dissociate and they diffuse through the plasma membrane, go and bind to other structures and activate them. So in this case, what will happen is the alpha subunit activates adenylate cyclase. This is an enzyme that's normally present in the plasma membrane. Okay, so once the alpha subunit binds to adenylate cyclase, that activates this enzyme. So now it's going to have an active site that will do something. And what it's going to do specifically is take an ATP molecule, an energy molecule, and strip off 2 of the phosphates and convert the remaining molecule into a cyclic form. So it becomes cyclic AMP or CAMP. CAMP then does something interesting. It goes over and binds to another molecule, and binding to this molecule causes the regulatory unit to fall off, and as a consequence of that, this protein kinase becomes active. So kinases, remember what kinases do. Kinases are a type of enzyme that phosphorylate other things. So now this protein kinase is able to phosphorylate and cause signaling cascades inside of the cell, and this is all through a second messenger system. In the end, there has to be a way to turn off this signal, and in the way that is done is by inactivating CAMP. And CAMP can be inactivated by this enzyme, phosphodiesterase. And just as an aside, this is one of the things that caffeine actually does if you drink coffee or tea or anything like that. One of the things that caffeine does is it inhibits this enzyme phosphodiesterase. So essentially caffeine is inhibiting the breakdown of CAMP. And so these signaling pathways stay active longer than they normally would. So that's just one example of what caffeine can do. Okay, next system is the Phospholipase C system. And this one, it's going to start off the same way. Epinephrine is the hormone that we're using here. So just before we get too confused, let me go back and forth between these slides a couple of times. Previous slide, okay, we said hormones epinephrine and norepinephrine. They use this CAMP system. Now on the next slide, we're saying epinephrine can use this phospholipase C system. So we have one hormone that can activate two different sorts of systems, and the difference is in the receptor. So which type of receptor is it binding? We've said that adrenergic receptors, like these, adrenergic receptors come in 2 varieties. There are alpha and beta types. So depending on which of those types are bound, a different system will be activated. So this gets really complex. On the previous slide, we were talking about beta adrenergic receptors. This was a beta adrenergic receptor. Now we're dealing with an alpha adrenergic receptor. So anyway, let's see here. Our hormone binds to its receptor, and that again is going to activate a G protein. This time the G protein activates something else. So this time, instead of adenylate cyclase, this time we're going to activate phospholipase C. And phospholipase C, once it is activated, it will use some of the membrane lipids and convert them into a couple of other forms. We're going to focus in on IP3. This is a molecule that is produced by phospholipase C. And IP3 will diffuse through the cytoplasm over to the endoplasmic reticulum. Remember, back when we talked about membrane potentials and calcium, we said that inside of the cell, calcium is kept in really low concentrations. So there are pumps that transport calcium out of the cell, and there are other pumps that transport calcium into the endoplasmic reticulum. That's a storage site for calcium. What IP3 does is it diffuses over to the endoplasmic reticulum and causes calcium to leave. Calcium can now come out of the endoplasmic reticulum. So this is going to temporarily raise the calcium concentrations inside of the cell in the cytoplasm. That's the phospholipase C system. Okay, 2 of 3 down. Let's look at the last one. The tyrosine kinase system. This is our last example of a second messenger system. Okay, tyrosine kinase. This system is a little bit different style. In this system, there are 2 receptor, well, each one is kind of like half a receptor. There are 2 molecules that are in the plasma membrane, and they're associated together. They're a dimer. Okay, the hormone molecule, insulin is the example we're using in this case. The hormone molecule comes and binds to the receptor, the dimer, and that binding initiates a phosphorylation event. And this is kind of strange. What's going to happen is this half of the dimer will phosphorylate the other one, and this half of the dimer will phosphorylate the one on the right. So they phosphorylate each other. This is called autophosphorylation. Okay, so now they both have a phosphate group. You can see those little red phosphate groups shown there. And once those phosphate groups are in place, this whole end of the receptor in purple, this whole end becomes active. And it is going to do some stuff. Okay, so this is going to act as a tyrosine kinase, and that will cause phosphorylation of signaling molecules, which are just shown in blue down here. The main takeaway is this whole end, once it's active, it is a kinase, and it will go and phosphorylate other things in the cytoplasm. And then that can set up a whole signaling cascade, again, inside of the cell. Okay, so with that was a huge amount of information that I just gave you. That was enough for a whole entire lecture. If we were in person together, that would be it for the day. So again, make sure to take some time to digest all of this. And what we're going to do next is go through specific glands of the endocrine system and see what each one does. So I think good news, the hard part is out of the way, going through these mechanisms. What's up next is going through some specific examples of different glands.